Sure. So we began to collaborate with Regina Barzilay and Tommy Giacola about 2 years ago. And really our idea was to see if we could harness the power out of our artificial intelligence to address the antibiotic resistance crisis. And together with John Stokes, a star postdoc in my lab, and a number of great graduate students in Regina and Tommy's group, we brought a team together that enabled us to do just that. We used AI to discover a number of new antibiotics, some with very potent activity. Regina and I were brought together as faculty co-leads for the new Jamil Clinic, which is MIT's new center focused on using AI to address challenges in healthcare. My lab had been working in antibiotics for over a decade, and Regina's group had moved into health applications for AI, but not from an antibiotic perspective. And we saw that we had mutual interests, overlapping interests, but differing expertises that when brought together could be used maybe in a very creative way to address one of the biggest crises we face in humankind, which is antibiotic resistance. I think it's important to be worried about it. I think resistance has been growing decade upon decade, largely due to overuse of antibiotics, and the overuse is compounded by reduced efforts to develop new and improved antibiotics. I think the way that we all can do a better job is by minimizing our use of antibiotics. Use them only in instances when you have a bacterial infection, not a viral infection, and where your doctor would prescribe the antibiotic is basically very, very needed in order to address the infection. I think we should reduce the use of antibacterials around our homes, and we should encourage the agricultural industry to significantly reduce the use of antibiotics as growth incentives for livestock. So in our effort with Regina and Tommy, we didn't use AI to design a new antibiotic, but instead use it to discover. And what features were taken into account were basically three, and that were the structure of each molecule that was used to train the model, the ability of that molecule to exhibit antibacterial activity, and the toxicity profile of that molecule. So we were able to train two different models, one around associating antibacterial activity with different features of the compound structure, and another associating compound structures with its possible toxicity. And using both could then arrive at predictions using it as a screening tool to identify novel antibiotics that we were then able to test and validate. We are looking to advance some, the most promising ones into clinical trials, and we've launched a nonprofit called FairBio, whose mission will be to take our most promising molecules and partner with biotech or pharma, as well as other nonprofits, nation states, and philanthropists in order to make sure we get these antibiotics into patients where they could be really helpful. I think the process downstream of what we're doing will not be very different from other efforts in antibiotics. The difference is really in the discovery efforts we have at MIT, where by training computers with a relatively small amount of training data, we arrive at a very powerful tool that can be applied to enormous libraries that would be impossible to screen experimentally, but we can screen them in a computer in a small number of days, opening up and expanding then chemical space that we can exploit to try to find new antibiotics. So this effort did not involve any collaborations with companies, and we are able to use databases that are available to the public. We firstly applied the model to a drug repurposing library that's available through the Broad Institute, where I run a lab. The Zinc database, which is the second large one we applied to, is available to all. And the challenge was then just, could we purchase or synthesize the most promising molecules that we identified in that library? And there were the challenges in potential time constraints, but we were able to identify 23 of the most promising ones that we could synthesize approaches that we were subsequently able to test. Yeah, so in fact, the rate is higher than you alluded, and maybe than I suggested. We were able to screen 107 million molecules in a matter of just 3 days, which is orders of magnitude faster than what can be accomplished experimentally. And frankly, from a pragmatic standpoint, it would be impossible to screen 107 million molecules, let alone compile 107 million molecules. It's just an enormous number. But we can do that, as I said, in just 3 days using the AI approach. And I'm sure we could get that time down even further. I'm not sure it'd make much of a difference if you're 3 versus 1, but we could, I think, reduce it even further. I think that artificial intelligence is going to become a useful tool as part of the drug discovery process. I think we're demonstrating already in antibiotics that it has value. I think we will continue to see advances in the antibiotic side, heading toward predictions for combinations, predictions for targets and mechanisms of action, shifting from a screening tool to also design tool. And the platform is not limited to antibiotics, and we think that the AI aspect in particular could be applied to other clinical indications, including viral illnesses such as SARS-CoV-2 infections, as well as complex conditions, complex diseases such as cancer and metabolic disorders. I think that I hope the young people who are watching this at MIT and across the country and across the world see that the pandemic has identified that we need young, motivated talent to move into infectious diseases. We have pressing problems, we have challenges, tractable problems that are of great import that could really benefit from innovation and young, motivated minds. So I hope that some of the listeners here will be motivated to consider this as a viable career path.